Hello everyone. Uh, Thanks for the Botany for You and its organizers for giving me this opportunity to talk about some basics about the drug discovery research and what are the challenges associated with and what are the opportunities. So, for any particular disease, there are several ways to manage the disease. It's called disease management strategies. It's has several arms, and the major arms are located shown here. And drug discovery is a key uh, strategy to tackle that particular disease. So it has several arms, as I told. So the main arms are strategic prevention. Strategic prevention is different from general prevention. Strategic prevention means vaccination or traveler's medicine, for example. So vaccination, we all know what is vaccination, but traveler's medicine, it's, for example, if we are going to a pandemic area, or endemic area, malaria, so doctor prescribe you to take some preventive drugs like mefloquine. So if you have that particular drug in your blood at very small amount, so when the malaria parasite will be exposed to the small amount, very small amount of the drug, it will be killed by it. Then precautions you have to take, and then general prevention means in this COVID situation, people say that use mask, don't go to the places where a huge crowd is there, just make the distance with each other. So these things are coming in general prevention category. Then surgery and other clinical procedures, we all know. Then basic and translational research. Uh, so researchers are working day and night to tackle the problem of disease and its associated problems. And then of course, patient care for the faster recovery of the patient who is suffering from some particular disease. And then drug discovery. So what is drug discovery? Drug discovery is comprehensive approach, and it involves several fields of the scientific research. So there are several approaches to develop new drugs. What they are: developing analogs of existing drugs. What does it mean? Means if a drug is already in clinical use, and for example, it has a huge level of drug resistance and patients are not cured properly with that particular drug. So then you have to make some changes in that particular moiety and then reintroduce into the system so that they can work. Like the best example is like macrolide antibacterial, like erythromycin. It's called first generation macrolide. So then when it has a resistance, then scientists develop azithromycin. They have made some changes in the erythromycin moiety, and then they make azithromycin, which is more efficacious than erythromycin. Then again, they develop clarithromycin. Then combination therapy. We will go details about the combination therapy. And in this particular corona era, this combination therapy is very important. We will go in detail in a couple of slides. Then identification of resistance reversal agent. As I already told you that there is a problem of drug resistance, which is a very big problem for drug discovery research and for the disease management. You must have heard about the MDR and XDR-TB, multi-drug resistant TB or multi-drug resistant malaria. So the parasite, it develops resistance against the particular drug which is in use. Then identification of new drug targets and synthesis of their selective inhibitors. We will also come to that. then analysis of compounds from natural products, and then exploration of drugs indicated for other diseases. You must have heard that this antimalarial is being used for corona treatment, like hydroxychloroquine. So this is the best example of exploration of drugs indicated for other diseases. Then coming to the overview of the drug discovery and development process. So there are two main phases of it. One is discovery, and the second one is development. So in discovery, you can categorize it as strategic research and exploratory research. So does it mean strategic research? First, we have to identify the target. So in target identification, we have to check the program selection. Program selection means which particular disease you are going to target, or which particular pathway of that particular disease you are going to target. So make a plan, then identify a target. I'll come to that, how to identify, and then validate that particular target, whether this target is very important for that disease or not. Then hit generation, so and lead identification. It's called, we can do it by virtual screening, by bioinformatics approach, or assay development and high throughput screening, HTS. We can design new pharmacophores, and then we can identify lead. Lead means a potential candidate for further development. So, and then and these things will be done in vitro condition or ex vivo condition. After that, in vitro and ex vivo means we are not using animals in this particular type of discovery, and then we should go for preclinical to select the drug candidate. So first we have to do identification of the efficacy of the drug in vivo system and its ADME profile. ADME means absorption, distribution, metabolism, and the excretion. And then we should go check its efficacy and the safety in animal model. It may be mouse, monkey, guinea pig, or other, or rat. And then after, if it clears, the molecule clears all the parameters in preclinical stage, then we have to file an application that this, but we have this data, we have this molecule, and it clears all the measures with parameters. Then we have to apply, and then the authorities will permit you for its clinical development. So the clinical development will be done in basically three phases: phase 1, phase 2, and phase 3. And then it will be approved for human use. So as I told, what are the rationale of the drug discovery? What is the target identification? How we do it? So we can analyze gene sequence, protein structure, and metabolic pathway. We have to identify which is more important in that particular type of disease or parasite. And then we have to validate that particular target by gene knockout or similarity studies. And we have to identify the lead by virtual screening, docking studies, or pharmacophore mapping. And then we have to optimize the lead in vitro by QSAR and QSPR studies or dynamic simulation studies by means of bioinformatics approach. Then we have to identify, do preclinical followed by clinical studies. So what is the clinical development? How generally we do. So here is the overview of it. So as we mentioned that we do preclinical studies in animals for acute toxicity, organ damage, dose dependence, metabolism, kinetics, toxicity, and carcinogenicity, mutagenicity. etc. So if it clears all the necessary parameters, then phase 1 clinical trial will be done in 20 to 50 healthy volunteers. I repeat, it's called healthy person to gather the preliminary data of the toxicity and its ADME profile. And then if it clears the phase 1, we have to go for phase 2. And the phase 2 will be done with the subjects with the disease, and it will be done in 150 to 300 subjects to determine safety and doses recommendation. And after clearance of the phase 2 parameters, it will go to phase 3, and it will be done in 250 to 4,000 more varied patient groups like males, females, different age groups to determine short-term safety and efficacy. And then it will be registered for clinical use. And after clinical use, there is a phase 4 surveillance. It's called phase 4, not trial, it's post-approval surveillance. So if the drug is going in the market, so we have to check its efficacy and its toxicity time to time. It's called phase 4. So how we develop the early drug, what is the overview of the process. So we identify, as I told you, the uh, target. So we have to select the pharmacophore from the different sources, and then we have to evaluate it in vitro against disease cell line. For example, if you are working on breast cancer, you have to take some breast cancer cell line, or directly some enzyme or particular target of that particular disease. Then you have to determine its efficacy by calculating IC50 data. IC50 means the concentration at which there is 50% inhibition. Then again, toxicity is a major issue in drug discovery process. So we have to evaluate its cytotoxicity against normal cells. And then we have, on the basis of its efficacy and toxicity in normal cells, we have to calculate the selectivity index. And if it crosses the a level which is approved for that particular disease, then we will go for in vivo preclinical and followed by clinical development. So there is a practical example of in vitro drug discovery. It's just a representation. So how to determine IC50 value? So first, the top left panel is showing a 96 pair plate. It's just a representative. You can use several other kind of plates recommended for different kind of assays. So you make a serial drug dilution of the drug in these plates, and then treat them with your, I mean, put your cells or your particular enzyme in that, and then incubate it for a recommended time. And then you have to add some detection probe, which is a chemical moiety or fluorescence labeled dye, and then you harvest it after a certain incubation, and then you detect the signal of that particular probe to determine the efficacy of the that particular pharmacophore, and make a graph and calculate the IC50 value. Similarly, you have to do the similar kind of assay in normal cell line to determine the 50% cytotoxicity. And then on the basis of these two data, you have to calculate the safety index of that particular pharmacophore. So as I told you, we will discuss in detail in combination therapy, because combination therapy is a very hot area during this COVID pandemic. So let's go for it. So combination therapy, why we do, covers more than one vital enzyme as their target to give better efficacy. It delays the development of the drug resistance against both the partner drugs. And if you use 2 or more drugs at a time, there will be shorter regimens which have better compliance. This means the less frequency of the drug gives you better compliance. So how you do in vitro drug combination? First, you assay the efficacy of drug A alone and the drug B or C alone, and then assay the efficacy of their combination, and then analyze the drug interaction by making an isobologram, and select the best combination for the drug. So how to determine? There is a formula. I'm not going to do very detailed, but how to calculate the um, synergy or antagonism or additive interaction of two or more drugs. So you have to calculate fractional inhibitory concentration of particular drug. So how to calculate? You have to calculate the IC50 of drug A in combination and divide it by IC50 of drug A alone. And then you have to calculate IC50 of drug B in combination. And then you have to calculate, divide it by IC50 of drug B alone. And then sum of this particular number, whatever you will get, and these two particular numbers, what you will get. So if the sum of these number is less than 1, it's a strong synergism, and it's 1, it's synergism, and if it's less than 2, it's additive interaction, and if it's more than 2, it's called antagonism. But these parameters, some people say that if 1 is additive and more than 1 is antagonistic. So it depends how, but of course, less than 1 is synergistic effect. Here is a typical isobologram. Here you see these dots, they show additive effect. And this particular one is slight synergistic effect because it's below 1. This is called isobologram representation for combination therapy. And after this, they have to go preclinical studies in mouse or monkey. Here is the representation of mouse. So what we do with the mouse, what is the use? We have to calculate the dose. We will get effect on our organ system. We will get the side effect of that particular molecule, and we will get the effectiveness, and we have to compare with the other drugs. For example, if the existing drug for that particular disease is more efficacious than the new one, then there is no use of this new moiety. So we have to compare with some standard drug, and it should be more efficacious than the standard drug. And the we will calculate the gender difference. It means whether it's working more in females or males, what are the differential effects in males and females. And then you will ask that how we go from mouse or monkey to human. So there is a formula. It's not very accurate, but at least we have something. So there is a formula given by Regan-Shaw et al. On the basis of that, we can calculate the dose from one animal to another animal. So how to calculate? There is based on the body weight and body surface area, and then it's calculated by the Km factor. So if you want to calculate effective dose in animal A from animal B, then it's equal to effective animal dose B in mg per kg into animal B Km divided by animal A Km. So this is the formula how we calculate, and as I already told, it depends on the choice of the clinician and other discussions also. This is not very accurate. Then in drug discovery research, pharmacology is a major branch, and it's a branch of medicine which is concerned with the use, effect, and mode of action of the drug. It covers the drug composition and the properties, synthesis and drug design, molecular and cellular mechanisms, organ system mechanisms, signal transduction and cellular communication, molecular diagnostics, drug interaction and chemical biology, therapy, and medical application. And the main two important areas of pharmacology are pharmacodynamics and pharmacokinetics. So here we will discuss, as I already told you, that toxicity is a major issue in drug discovery process and how pharmacodynamics deals with it. So there is a word called pharmacovigilance. So what is pharmacovigilance? WHO says that the science and the activities related to the detection, assessment, understanding, and prevention of adverse effects or any other drug related problem is called pharmacovigilance. And this applies throughout the life cycle of a medicine, equally to the pre-approval stage as to the post-approval. So what is the need for the pharmacovigilance? So there is a word, there is a sentence, I'm quoting somebody. So it said to undergo treatment, you have to be very healthy because apart from your sickness, you have to withstand with the medicine. So what is the risk of the toxicity of that particular drug? So WHO says that no medicinal or medicinal product is entirely or absolutely safe for all people in all places at all times. We must always live with some measure of the uncertainty. So here are some media reports which says that why this assessment of the toxicity is important. Artesunate aminoquine is the first line antimalarial treatment, but there are several reports that it has a major toxicity issue. Again, and so the safety concern is now high on agenda of all countries, even developing countries, or there are some reports from the developed countries also. So it's this is a major concern throughout the globe. So how to tackle this problem? So we have to be very cautious about the animal experiments and clinical trials. We have to do epidemiological methods like spontaneous reporting. We have to do cohort event monitoring or phase 4 studies. So and then case series, and we have to have registration of the patients and record linkage, and then after several reports of the clinical trials, we have to do meta-analysis of these clinical trials and come to a conclusion that whether this particular drug is doing some harm to the population or a particular population in a certain area, and then we have to tackle this problem. So coming to the COVID-19, because I jumped suddenly to the COVID-19 because it's a very hot area these days and very concerned to the whole world for the mankind these days. So as I think most of you must have heard about its life cycle, it goes through its receptor like ACE2, which is angiotensin receptor, or TMPRSS2. TMPRSS2 is androgen-driven gene and protein. Its product is protein, and then it comes to the cell, and then it propagates, and then it lies the cell membrane, and then goes out and infects next cell. So as I told, why I mentioned the drug or combination is more important these days, because there are several reports that in absence of vaccine, only combination therapy is working in case of COVID-19 patients. So here are some examples, and I just took a snapshot from different sources. So what are the future perspective for, especially for COVID-19, that angiotensin 1 converting enzyme, that called ACE2, it's an important component of the angiotensin system, which provides an entryway of coronavirus into the human cells, and delineating the relation between the hypertension and coronavirus infection. Recent correspondence, one of the good journal called Lancet, suggests that coronavirus enters the cell via binding its S protein on ACE2 receptor. So, the, a potential therapy for coronavirus is selectively inhibiting ACE2 without disturbing any other component of the renin-angiotensin system. With combination of other potential candidates would be a better option. But as of now, what I heard or read that there is no specific angiotensin receptor 2 inhibitor is available for clinical use. There is one that which is not approved yet, which is very specific to ACE2, not the other ACEs. So uh, if there is some particular ACE2 inhibitor which is not inhibiting other and other uh, uh, angiotensin receptors, that would be a better way to tackle this COVID-19 situation in absence of the vaccine. So, in conclusion, uh, combination therapy followed by the drug interaction analysis would be a better tool for future drug discovery. And thank you all. I'll be happy to answer questions. You can follow me or you can mail me. And thanks again.